All right, so this is histology. Specifically, we are talking about histological techniques. Now, anatomy has got three divisions. We have got gross anatomy, histology, and embryology. In gross anatomy, the first topic that we discussed was the anatomical terms and the language of anatomy. In in histology, the first topic is histological techniques. In embryology, we start first talking about cells. We move from cells to how sperms and ovums will develop, and then we'll start now going like that. What happens? Fertilization, and then the baby starts to grow. So, histological techniques. First thing first, what is histology? Histology is the study of the tissue of the body and how the tissues are arranged to make up an organ. Actually, the word histo, coming from Greek, means tissue or web. So you know these tissues are a lot of cells put together, and then they end up forming now an organ. So tissues are webs of interwoven filaments and fibers. So you have got cells and non-cellular components, which makes up what we call a tissue. This is important for you to know because all the types of tissue, when you are trying to describe them, you need first to know what are the cells that make up this tissue. Number two, what are the non-cellular components of that tissue? And we call that as extracellular matrix. Okay. Histology is the aspect of tissue biology which focuses on how the cells structure and arrangement optimize function specific to each organ. How is it that this organ specifically does this function? It's because each organ has got its own cells and they have been set in such a way that they only perform that function. Now, as I said, each tissue is made up of two things: a cell and the extracellular matrix. Think of like a house. A house is made up of bricks and what holds the bricks. So the extracellular matrix is what holds the bricks. Now, of the extracellular matrix, you have got two things. You have got protein fibers and ground substance. For these protein fibers, they include the three types of proteins. You have got collagen, which is the most abundant in most types of tissue. You have got reticular fibers and elastic fibers. So we are going to see when it comes to different types of tissue, the components, the composition of these three types of fibers is going to differ. Some tissues are going to have more collagen than reticular, others are going to have more elastic than reticular or collagen. So that will simply describe the function of that tissue. For example, tissues that have got collagen fibers are mostly strong tissues. Examples we can give are bones, we have got tendons, ligaments. So those are strong because collagen is strong. And then for things that are a bit soft, they are going to have reticular and elastic tissue to be a lot. Now, besides the fibers, you also have what we call ground substance. Okay, so think of it. I have said that cells are like bricks, and then extracellular matrix is like what was the bricks. So, of what was the bricks, remember when they are making a house, there are these wires that they use. So, wires, think of them as protein fibers to hold the bricks, but you don't just put the wires alone. Okay, what other thing do you do? You also have water and mud. So, that water and that mud will actually make up what we are calling as ground substance, and that is the component of tissue. So we have got three many types of tissue. Actually, all the cells in our body, as many as they are all going to just be made up of these three types of tissue. Okay, I mean these four types of tissue. So you have got nervous tissue, epithelial tissue, you have got connective tissue, and muscle tissue. And one thing that I want you to know of this is that what is the composition of the extracellular matrix for each one of these types of tissue? So you can see that the nervous tissue has got only a very small amount of extracellular matrix. Okay. And then the epithelial tissue has got a small amount also, but not as small as compared to nervous tissue. Muscle tissue, it is just a good amount of extracellular matrix. Connective tissue is nearly entirely made up of extracellular matrix. So it's important for you to know this. Very important for you to understand that. You are going to find some questions where they are going to ask you that. Please make sure that you know connective tissue is the tissue that has got the most abundant extracellular matrix. What are the functions? What is the function of the nervous tissue? The nervous tissue is found in the nervous system. The nervous system is involved in conduction of impulses. The reason as to why you are able to feel pain is because of nerves. And then being able to coordinate the entire body is because of the same nerves. And then the epithelial tissue is going to line body cavities. So lining the body, that is the epithelial tissue. For example, you have got your skin. The skin is made up of epithelial tissue. To just a second. Okay. So we are saying we have got four main types of tissue. The nervous tissue is going to be involved in transmission of impulses in the body. For example, for me to feel pain, for me to move my muscles, I need nerves. Without a nerve, you can't feel anything. And then epithelial tissue is to line body cavities. For example, the entire body is covered by epithelium. The skin, the GIT, every place where you see an hollow opening, like an opening, the GIT, the respiratory system, the reproductive system, every surface of the body is going to have epithelium. We'll discuss the two types of epithelium later on as is a topic. And then the muscle is involved in contraction to produce movement. And then I've got connective tissue. Connective tissue is just there to support and protect organs or other tissues. Okay, think of it, you have got this organ. The organ is going to have different cells, and then for you to connect the entire body all together, you need connective tissue. So even when we have got so many cells in our body, the entire body has been divided into only four types of tissue. But now the way you make up the tissues, first you have got cells. Group of cells coming together, they make up tissue. Group of tissues coming together, they make up an organ. Organs coming together, they make up a system, and that makes a lot of systems. In our body, we have got 11 systems. Those will make up an organism. We've talked about the four types of tissue. Okay, so if you want to understand the human body, you are supposed to study a tissue. And for you to study a tissue, you need first to collect that tissue. For example, let's say someone says they have got a cancer. How do uh, how do you know that someone has a cancer? What you do is that you are going to get a sample of the body. You basically get a sample of the cells or the tissue. And then once you get that sample, or for example, let's say someone was involved in an accident or something, and then a part of the body was cut. You can get that part that has been cut, okay, and then you look at it on the microscope. So now, what are the steps it takes for you to prepare a tissue in order for you to study it on a microscope? So most tissues and organs, when you get them, for example, when you collect a sample, a specimen from the body, you want to study it, they are too thick, they are very big, you can't study them on a microscope. So what should you do? You have to cut them into thin specimens. Those specimens, you are going to cut them using an instrument which you call a microtome, and then you are going to now put it on a slide to view under a microscope. Okay. So this instrument that you can see here is known as a microtome. The microtome is going to have a point that you are going to put a tissue, and then the tissue is going to be cut into very small objects. I mean small specimen. You need to know how this one is looking because it can come in your histology practical test. Identify the instrument. When you see this instrument, it is a microtome. What is its function? Is to cut tissue into thin specimen that you can view under a microscope. So these are the stages for tissue preparation. You need to know the stages in order. Okay, you can see the way they are going like that. So what are the steps of tissue preparation or the stages? The first thing that you do is you first obtain a fresh specimen. You are getting this specimen from the animal that you want to study or the human body, right? Once you get that specimen, you don't let it to be like that because it can get destroyed. So what you do is that you preserve the tissue. And the preservation of tissue is known as fixation. They are actually what we call fixatives or solutions that you are going to use to preserve a tissue. For example, you hear of formalin. For you to preserve a dead body, you use formalin. And then after you've preserved that tissue, the next thing that happens is to remove water from that tissue. That is known as dehydration. After you remove water, you are now going to infiltrate that tissue with paraffin, and that is known as clearing. And then what happens next is that you are going to infiltrate the paraffin wax. The first one was a solvent. This paraffin solvent was to remove the water. That was clearing. To clear is to remove. And then now you are going to infiltrate with paraffin wax. You want to like harden the tissue. That is known as embedding. And then you now start cutting this tissue into smaller pieces. That is the known as trimming. Okay. And then after that, you are now going to in trimming you are putting the paraffin block in orderly condition by clipping or pruning. It's a very big tissue, and then you are just cutting it into at least nice size, but cutting into very thin specimen using a microtome. That's known as sectioning. And then after you section, you've cut into very small objects. You are now going to stain your tissue so that you can be able to see the cells, because the components of the cells stain different colors with different stains on the microscope. After you do that, now you're going to get your tissue and put it under the microscope so that you can view it. So these are the steps which we need to know. Now, just talking about them in details. So I'll start with fixation. At this time, we are saying we've already obtained our specimen. So how do you preserve the specimen that you've obtained? So in fixation, what you are simply trying to do is to stop that tissue that you have collected from undergoing degradation or autolysis, being eaten up, because tissues have got their own enzymes and the enzymes are capable of eating them up. So to prevent autolysis and to preserve the cells and the tissue, that's why you do fixation. Now, the substance that we use in fixation to protect the tissue from degradation, we call it a fixative. This fixative should diffuse through the tissue in order to preserve the tissue. So, in order to do that, what happens is that the specimen has to be cut into smaller fragments, okay, to facilitate the penetration of the fixative. Intravascular perfusion or fixative can be used. You know, the meaning of intravascular perfusion, intravascular, it is within the blood vessels. So what you can do is you can simply also get your preservative or your fixative, put it into the blood vessels, and then using your veins, and then it will be able to diffuse through the tissues or the organs. Okay. Because the fixative in this case rapidly reaches the tissue through the blood vessel, fixation is improved. So you can as well, when you use blood vessels to take your preservative, for example, if someone is dead, just inject formalin through the blood vessels. In anatomy later on, we are going to learn which specific blood vessels are you supposed to, actually I want you to find out. Go and do a simple research. Which blood vessels do we use for in putting formalin in the body in fixation? All right. So, what are the examples of fixatives? What are some of the fixatives we can use to preserve tissue specimen? We can, for if you want to study that tissue using a light microscope, you can use formalin. Now, formalin is an isotonic solution, meaning that it has got the same concentration as the concentration inside the cells. It is actually 37% formaldehyde. Organic chemistry. We'll discuss. You understand the structure for formaldehyde. But just take note that it's 37, formalin is 37% formaldehyde. Please take note, you use formalin for light microscope. And then if you want to use an electron microscope to study that tissue, you are going to use glutaraldehyde. Don't forget this, all right? So both formaldehyde and glutaraldehyde will be able to react with the amine group that is present in the proteins of the tissue. And that is what prevents the tissue from undergoing degradation. Now, with a greater magnification and resolution of very small structures in the electron microscope, fixation must be done carefully to preserve ultrastructural details. What we are saying here is that when you are trying to preserve the tissue, as you are trying to preserve the tissue, what happens is that you can end up destroying the tissue. And so, as you do the preservation, you need to make sure that you don't destroy. Okay, you don't destroy the small parts of the cell. You know, a cell is the smallest part of a living organism, but a cell also has got smaller things inside it, the organelles, and much more smaller things also inside the organelles. So, I want to make sure that you preserve all those things. So, towards the end, a double fixation procedure using a buffered glutaraldehyde solution followed by immersion in buffered osmium tetroxide is a standard method to prepare tissue for such studies. If you want to prepare to preserve these ultrastructural details, what you do is you do double fixation. You are going to use a buffer solution of glutaraldehyde and then you immerse it in osmium tetroxide. So osmium tetroxide preserves and is actually also a stain. Osmium stains the lipids. You can find this in your exam. What stain can we use for lipids? Osmium tetroxide and also proteins. So remember, after you have preserved your tissue, the next thing that you need to do is dehydrate to remove water from that tissue. So in dehydration, what happens is that water is extracted from the fixed tissue by successive transfer through a graded series of ethanol. So in other words, when you want to remove water from the tissue, you use alcohol, and that alcohol is ethanol. And how do you do that? You are going to be adding ethanol to the tissue in an increasing order, starting from 70%. Think of it when you have got a sponge, right? Or say you are washing your clothes. You want now to hang them. What you do is you are going to start compressing them so that you remove the water. At the beginning, you first compress, not so much strong. You remove water. At the next time you compress, you do it much stronger. Again, when you do it again, you are going to even compress even much stronger because you want to remove even this little water that has remained there. So that is why we use this percentage of ethanol. You start with 70%, 70% to remove some water, you go to 80%, you go to 90% alcohol, and then you go to 100% alcohol. That is how you remove the water. Hope this makes sense. Please, if there's a question, be free to ask at any point. So once you've removed the water, the next thing, remember you've removed water, but you've put in what? You've put in alcohol in your cell. So the next thing should be clearing. Clearing is you getting to remove that ethanol. And how do you remove the ethanol? You are going to replace it by an organic solvent. Now, this organic solvent that you are using is miscible with both alcohol and the embedding medium. Now, remember the previous step before clearing was dehydration. In dehydration, we are putting alcohol. The next step after clearing will be embedding. So what you want to do is during clearing, you want to use a solvent, organic solvent, that can dissolve or mix with the solvent that was used in the previously step, which is in uh, in removing of the water dehydration, and the same solvent should also mix with the solvent that you are going to use in the next step, which is embedding. Hope that's clear. Okay, so as this solvent that you are going to be using enters the tissue, they are going to become more transparent. That's why we call this as clearing, because once you are removing this alcohol and then the organic solvent is replacing entering the tissue, it becomes clear, and that is why we call this as clearing. The fully cleared tissue is now placed in melted paraffin in an oven. Please take note of the temperature at 52 degrees to 60 degrees. That is going to be the next step. Please take note that the tissue that has been cleared, you now put it in melted paraffin. The paraffin is at a high temperature, 52 to 60 degrees. That's why it is melted. And so that now takes us to embedding. So embedding, the tissue are embedded in a solid medium to facilitate sectioning. Embedding means you are trying to make sure that a solid penetrates, enters the tissue, and then so that it can be easy for that tissue to be cut, because when you get a tissue from the body, it is soft. You can't cut or deal with something that is soft. So in order to cut very thin sections, tissue must be infiltrated after fixation with embedding material that imparts a rigid consistency. You want to make your tissue to be strong. So embedding materials, what are the examples of materials that we use? Paraffin. Okay, we've already talked about it in the previous. Remember, you used melted paraffin at this temperature. That was embedding. So for light microscopy, you can use paraffin and resins. And then for electron microscopy, you can only use the resins there. Of course, there can be other things that you can use. So the fully cleared tissue, we said it is placed in melted paraffin at 52 to 60 degrees. At such temperatures, the clearing solvent, what happens that the solvent evaporates, and then the tissue is going to be now filled with this same paraffin. Okay. The impregnated tissue, the tissue that has been filled with the paraffin that is liquid because it is at high temperature, is now then going to harden in a small container of paraffin at room temperature. So in other words, you can see the temperature is high here. Now you are trying to lower the temperature. When you start lowering the temperature, the paraffin that was liquid will start becoming solid, and so it will end up making the organ or the tissue to be hard. Okay, so the ethanol solvent are going to be replaced by plastic solution, which of course can be the paraffin as it gets to become solid, and this is done with addition of cross-linking polymerizers. So it's like the embedding material that you are using is connecting these tissues, connecting to one and another like that to become strong. Plastic embedding avoids the higher temperature needed for paraffin embedding, which helps avoid shrinkage and major distortion of the tissue. So embedding, you don't only use paraffin, you can also use plastic. And for plastic, it will avoid high temperatures. And because those high temperatures can actually distort, can destroy some components of the tissue. So after the embedding, the next thing will be sectioning. Okay. The next thing is going to be sectioning. It's after embedding that we cut this into smaller portions. That's known as trimming. Now, to now make much thinner slides, that's now known as sectioning. So in sectioning, a hardened block containing tissue and paraffin is placed on a microtome, and then it's going to be sliced, because a microtome has got a blade, it has got a laser, okay, into extremely thin sections. Paraffin sections are going to be cut at 1 to 10 micrometers thickness. Can you see that? That is very small. A micrometer, that is very thin, very thin. Thinner than a paper that we use for writing. Okay? So The glass or diamond knife of the ultra microtome can produce sections of less than 1 microtome for electron microscopy. That's for an ultra microtome. You can see it can even have much smaller sections. So very thin sections are placed on a glass slide and then stands for microscopy, or on a special grid for electron microscopic staining and examination. So the next thing, after you've sectioned into very, very small parts, now you do your staining. Okay, staining is done using basic and acidic dyes to be able to stain the components of the cells. Why do we use basic and acidic dyes? Because nearly all the components of the cell are either basic or acidic. So the tissue components such as acidic components. So things that are acidic actually have got a negative charge. What are the examples of acidic components of the tissue which have got a negative charge? The nucleus. Basically, anything that has got nucleic acid is acidic because it's going to have. Remember the phosphate of a nucleic acid? That phosphate is negatively charged. So examples of such structures which are acidic, we have got DNA because they've got nucleic acid, RNA, and glycosaminoglycans. If you remember from our biochemistry, we did discuss the glycosaminoglycans. And then also the matrix of cartilage. Please take note the matrix of cartilage. And then you have got also the basic components of the tissue. The basic components of the tissue, these ones, think of the B for blue. So the basic components will stain blue or purple. While the acidic components, these ones normally stain pink. Okay, so the structures that are acidic will be stained by basic dyes. Please take note of this. While basic structures will be stained by acidic dyes. So the acidic structures we have understood here is DNA, RNA, glycosaminoglycans, and the matrix of cartilage. Basically, DNA is found in the nucleus. So we are simply saying that the nucleus is acidic. So it will be stained by basic dyes. You need to know examples of basic dyes. The most common one is hematoxylin. And then most of them, the basic dyes, just think of B and blue, B. So most of the basic dyes end with a blue. For example, toluidine blue, methylene blue, Gaussian blue. If you see those with blue, just know that those are basic dyes. They will stain the acidic components of the cell. And then those components of the cell that are positive, they are basic. And they're going to be stained by acidic dyes. What are the examples of structures that are basic? The cytoplasm has got proteins, which have got many of these positive charges. Okay? So the mitochondria, secretory granules, and collagen. Please take note. These guys are going to stain pink because they are basic, and basic structures stain with acidic dyes. Acidic dyes are pink or red in color, most of them. Example, the most common one is eosin. You also have orange G and acid fusin. These are acidic dyes. So normally when you are staining a cell, you actually combine hematoxylin and eosin. So we call it the H and E stain. The H is going to be staining the DNA, RNA, and these other structures. The E is going to be staining the mitochondria, the collagen, and these other parts. Okay. So in the H and E staining, the main stain here is a hematoxylin, which is the H. Eosin is known as a counter stain. Like those parts that can't be stained by the hematoxylin can now be stained by the eosin. So eosin is the uh, counter stain to hematoxylin. And then the final step before microscopic observation is mounting a protective glass cover slip on the slide. So once you've stained, now you get your specimen, you put it now under the microscope, and then you put a cover slip on top to be able to view under the microscope. Now you have got other dyes that you need to understand. The way questions come is they can mention a dye and then they tell you if it stains this cell, what color is it going to stain? Or they'll just say what stain was used. They will give you a slide. What stain was used. So you have got what we call the periodic acid Schiff, which is PAS. PAS, this one stains glycogen, glycoproteins, glycolipids. Okay, basically to stain polysaccharides and mucosubstances, substances that have got some carbohydrate in them, glycoprotein, glycolipid, mucin, like this is mucus. Mucus is also having carbohydrate. So the color that it stains this, the glycogen and these others, is deep red, also known as magenta. So that is for PAS. You also have the Mallory staining technique. So please take note. If they say Mallory staining technique, the Mallory staining has got three dyes. You have got Gaussian blue, acid fusin, and orange G. You see that in equation. What are the components of the Mallory staining technique? AAO, Gaussian blue, acid fusin, and orange G. So the Mallory staining technique will stain collagen, cytoplasm, and the red blood cells. You also have the Gomori trichrome, which will be able to stain the muscle, the nuclei, the collagen, and then you have got the Verhoef stain. Please, this one, don't forget it, the Verhoef. Remember, I said all tissue is made up of cells and extracellular matrix. Extracellular matrix is made up of protein substances and ground protein fibers and ground substances. And then protein fibers, I mentioned three. You have got collagen fibers, you have got reticular fibers, and elastic fibers. So the special stain for elastic fibers or elastic tissue is the Verhoef stain, and it stains black as you can see here. Okay. And then you have got this one, you also need to know it, the James stain. The James stain is also known as the right stain. This one is actually the main stain that you use for all blood cells. So blood cells, you use the James stain. What are the components of the James stain? You have got azu, stain, methylene blue, and eucin dye. These are the three components. And it's a special stain for all blood cells. So this table is just telling you of some stains and what they can stain and the color. Okay, sometimes anatomy can be a bit weird. They'll just say if you use the Mason trichrome, what color does it stain the nucleus? You can say that's blue. So you can just try to check up that. I've also written them properly here. Don't forget collagen fibers are stained by Van Gieson. Okay, and the Mason trichrome. Elastic fibers, Verhoef. Reticular fibers, you have got the silver. So we call this a silver impregnation or the silver salt. So every time you hear of silver salt, think of reticular fibers. Elastic fibers, Verhoef. Collagen, Van Gieson. And then now you can know also cell substance. This one is found in neurons. It's actually the rough endoplasmic reticulum in neurons. It was then using the Cresio fast violent. Melanin, which is the black pigment of our skin, stands with Mason Fontana. Lipids, as we said earlier, they stand with Sudan black osmium, osmium tetroxide. Okay, at least these ones, make sure you know them. So once you've done your preparation of tissue, what remains is now for you to get that tissue and put it under a microscope. But for a microscope, we are going to discuss that in the next class. And what I would just love you to do is to look at some of these exam questions that I put here. Okay? Some of them are on a microscope, but for a microscope, we'll do it later. Collagen fibers are, you need to state, are they basophilic, acidophilic, metachromatic, or bifringent? And then you also which of the following staining procedures relies on the cationic and anionic property of the material to be stained? Cellular storage deposit of glycogen, a free polysaccharide, could best be detected histologically using one procedure. So what procedure can we use to stain glycogen? All are basic dyes except, just like that, your test one or your final exam. Okay. One of the following is a basophilic tissue structure. So get one there. Which of the following stains blue with H and E? Smooth muscle can be best differentiated from collagen by, choose the correct statement. In hematoxin and eosin staining, hematoxin is a counter stain. Reticular fibers are best stained by silver stain. Basolamina is best stained by H and E. In H and E, nuclei appear blue. Okay, can also look at that. What is right stain used primarily for blood, fat, nervous tissue, elastic fibers? What color do elastic fibers stain with Verhoff elastic stain? During the preparation of routine H&E slide, what allows the tissue to be visualized? Which of the following would be best suited to visualize reticular fibers? During the preparation of routine H&E slide, what step occurs after the tissue is preserved? True or false? But this one is on a microscope. We'll look at that. The following is best stained with hematoxylin. All are basic dyes except the following dyes are constitute the James stain. You can check which one is known, which one is the answer. Glycogen is best stained by, which of the following technique stains the lipid in myelin sheath of nerves black? The following is the ascending order of tissue preparation steps in histology. Start from the first, going in the order that we've discussed them. Tissue from the following organs are best stained in silver stain except. So you need to remember silver stain stains reticular fibers or reticular tissue. So you are basically seeing which of these guys have got a lot of reticular tissue. So that is it for this class. Thank you so much for being present.